Hello everyone, this is the chapter 8 of level 2. Here we will discuss about absorption, diffusion, osmosis, imbibition, plasmolysis, water potential, transpiration and guttation. So absorption, transport and water loss in plants. Water is the most important component of living cells. It enters the plants through roots and then moves to other parts and is also lost by transpiration through the leaves. There are several phenomena involved in the movement of water about which you will study this lesson. Absorption of water. Just watch this animation. How water absorbed by plants. So here, apart from drawing water up by the xylem vessels, through transpiration pull also help the absorption of water by the roots. You can see in the root, water in the cortex cells near the xylem vessels enters the xylem vessels due to the transpiration pull. This decreases the water potential of the cytoplasm of the cortex cells. Water moves from the neighboring cells of these cells by osmosis. As a result, a water potential gradient is set up across the cortex, so causing water to move inward from cell to cell by osmosis. Some water moves inward along the cell wall. As water is drawn into the inner cells, the water potential of the root hair cell become lower. The water potential of the cell is now higher than those of the root hair cells. Therefore, water in the soil enters the root hairs by osmosis. This is how water is absorbed into the root. So after completing this lesson, you will be able to define the terms permeability, diffusion, osmosis and plasmolysis. Define and differentiate between the active and passive absorption. Explain imbibition, water potential, trigger pressure, wall pressure, wilting. Describe the pathways of water from root hair up to leaf. Describe the mechanism of translocation of solutes in plants. Explain the process and significance of transpiration. List the factors affecting the rate of transpiration. Explain the opening and closing mechanism of stomata, potassium ions theory, and list the factors affecting stomatal movement. Explain the process of guttation and list the factors affecting the guttation. 4 basic phenomenon: permeability, diffusion, osmosis, and plasmolysis. So permeability means this is the property of a membrane to allow the passage of the substances through it. The plant cell wall is permeable because it allows both solvent and solute molecules to pass through it. Cuticle layer is impermeable. All biological membranes like cell membrane, mitochondrial membrane, nuclear membrane, etc. are selectively permeable as they allow penetration of only solvent molecules but not the solute molecules. Diffusion. If a can of volatile substance such as ethyl ether is opened in a room, their molecules will soon be distributed until their concentration is the same throughout the room. In other words, ether molecules diffuse into the air in the room. Similarly, the fragrance of incense stick or agarbatti spreads from one corner of the room to the other due to diffusion. Another example is placing a small crystal of a water-soluble dye, copper sulfate, at the bottom of a test tube and then pouring water carefully over the crystal. Dye molecules will dissolve and the color will spread slowly throughout water, partly because of the movement of dye molecules through the water and partly because of the movement of water molecules in the region close to the crystal. Diffusion is the intermingling of molecules of the same or different species as a result of their random movement. It is dependent on the difference in concentration in the adjacent areas, and this difference is called diffusion gradient. Diffusion is an effective method of transport of matter over short distances. For diffusion to take place, no membrane is required. If a membrane is present, it should be fully permeable. The cell membrane are permeable to both gases CO2 and O2, and hence the two gases are able to diffuse. You can see this in this figure: copper sulfate crystals and diffusion into water. Diffusion, osmosis. So what is the difference? You can see this in this animation. Number one is diffusion. So diffusion, movement of solute molecules from an area of higher concentration to an area of lower concentration. Remember, movement of solute molecules, only solute molecules. Next one is osmosis. In the osmosis, semi-permeable membrane is required. So osmosis is the diffusion of solvent across a membrane, means movement of solvent molecules, only solvent molecules. Remember, from a region of lower solute concentration to a region of higher solute concentration. Osmosis. It can be regarded as a special kind of diffusion of water molecules from a region of their high concentration to their region of low concentration through a semi-permeable membrane. In osmosis, the water molecules move, and the presence of a semi-permeable membrane is essential. Experiment to demonstrate osmosis. Experiment to demonstrate the phenomenon of osmosis through plant membrane with the help of potato osmoscope. Requirement: a large potato tuber, 10% sugar solution, beaker, water, scalpel, pin. Method: take a large potato tuber and peel off its outer skin with the help of scalpel. Cut its one end to make the base flat. Now make a deep hollow cavity on the opposite side. Pour some sugar solution to fill half of the cavity and mark the level. By inserting a pin in the wall of the tuber, put the potato in the beaker containing a small amount of water and allow the apparatus to stand for some time. Make sure that the level of water is below the level of potato. Observation and conclusion: the level of sugar solution in the cavity rises. It is because of the movement of water molecules into the cavity from pure water in the beaker. This experiment shows the phenomenon of osmosis. You can see this in the figure. So explanation: the living cells of potato tuber collectively act as differentially permeable membrane. It means membrane which permits movement of certain molecules only through it. The two solutions, pure water in the beaker and sugar solution in the cavity, are separated by living cells of potato. Water molecules continues to move through the membrane into the sugar solution till the concentration become equal. In sugar solution is taken in the beaker and pure water in the cavity. The result will be reversed. The movement of water will not occur if the skin of potato is not removed, because the skin acts as impermeable layer. Just see this diagram. So diffusion and osmosis, difference between them. Diffusion is a movement of a given substance from the place of its higher concentration to an area of its lesser concentration, presence of semi-permeable membrane is not required. But on osmosis is a special type of diffusion of solvent molecules such as water from lower concentration to the higher concentration of solution when the two are separated by a semi-permeable membrane. Diffusion. The diffusion may occur in any medium. The moving particles may be solid, liquid or gas. In osmosis, it occurs in liquid medium and only the solvent molecules such as water move from one place. If you place a cell in a solution, it may shrink, swell or remain unchanged on the basis of relative concentration of water and solutes with respect to their concentration in the cell. Isotonic. The solution has the same concentration of water and solute as inside a cell. Cell remains stable in isotonic solution or there is no entry or exit of water from the cell. Hypotonic. The solution outside has lower solute concentration than inside the cell, so the cell swell as water enters the cell. Hypertonic. The solution outside has higher solute concentration than inside the cell, so water from cell moves out to the protoplasm of the cell. The cell shrinks and collects in the cell. So osmotic pressure and osmotic potential. When pure water is separated from a solution by a semi-permeable membrane, pure water tends to enter the solution by osmosis. Now the pressure required to prevent the osmotic entry of a water in a solution is called osmotic pressure. Imbibition. Before cooking chickpea or gram, it is soaked in water overnight. Next morning, the dry chickpea looks well swollen as it has imbibed water. Imbibition in plant cells refers to the absorption and adsorption of water by protoplasmic and cell wall constituents. Water is absorbed as a result of both diffusion and capillary action. Imbibition is a process that accounts only when solid plant material, dry wood, dead or living air-dried seeds comes in contact with water. In case of living dry seeds, water is initially absorbed by imbibition and thereafter water is absorbed by osmosis. Imbibition produces a large pressure, so much so that dry wood can even break a piece of rock in the presence of water. Because of imbibition, the wooden door during rainy season swell up and it becomes difficult to close the door. Importance of imbibition. Imbibition is the initial step in the germination of seeds. It causes swelling of seeds and breaking of seed coat. Plasmolysis. When a cell is placed in a solution, it will either shrink, swell, or will remain unchanged depending upon the concentration of the bathing solution or the solution in which the cell is placed. When a cell is placed in a hypertonic solution, when the concentration of the outer solution is higher than the cell shape, water from the cell move out resulting in shrinkage of the protoplasm in the center of the cell and disappearances of the vacuole. This phenomenon is known as plasmolysis. The space between the cell wall and the protoplast is occupied by the bathing solution as of the cell wall is permeable. When such a plasmolyte cell is placed in a hypotonic or dilute solution or pure water, water moves into the cell, causing the protoplasm to stretch and get back to its original shape. This phenomenon is known as deplasmolysis. The cell becomes fully turgid. So watch this animation. This is the example of imbibition. The adsorption of water by hydrophilic compounds is called imbibition. In this process, water molecules get tightly absorbed on the surface of compounds without forming a solution. Due to imbibition, these compounds show swelling. It is a common observation that during rainy season, wooden doors and windows imbibe water and swells. The hydrophilic compound like cellulose and pectic compounds in root hair cell imbibe water. So thus imbibition is the first physical process involved in the absorption of water. When a cell is placed in an isotonic solution or a solution with similar concentration as the cells have, there is no change in the shape of the protoplasm or the cell. Plasmolysis is a physical phenomenon. A cell can become plasmolysed and deplasmolysed depending upon the outer solution in which the cell is placed. No chemical change is caused due to the cell. Plasmolysis is a kind of defense mechanism against adverse conditions such as hyperthermic solution. You can see this in the figure, example of plasmolysis. So water potential or chemical potential of water is the energy of water molecules or tendency of water to leave a system or the ability of free water molecules to do work or move. Water moves from a region of higher water potential to a region of low water potential. Potential of pure water is taken as zero. When solutes are dissolved in pure water or in a solution, some water molecules are used in dissolving the solutes, thus less number of the water molecules are available to do the work. Hence, a solution has less energy or potential as compared to pure water. The water potential of a dilute solution is more than that of a concentrated solution. The value of water potential of a solution is less than that of pure water or zero. A negative number, water potential designated by a Greek letter psi. Pure water has highest water potential. It means 0 for pure water. Water potential determines the water status in plant cells and tissues. The lower the water potential in a plant cell or tissue, the greater is its ability to absorb water. Conversely, the higher the water potential, the greater is the ability of the tissue to supply water to other more desiccated cells or tissues. Turgor pressure is the pressure exerted by the protoplasm against the cell wall. The turgor pressure is equal to the back pressure exerted by the cell wall against the protoplasm. This back pressure exerted by the cell wall is called wall pressure. These two pressures are equal and opposite in direction. When turgor pressure becomes more than the wall pressure, the cell wall will burst. Turgor pressure is maximum when the cell wall cannot stretch anymore. Such a cell is said to be fully turgid. At this point, a dynamic equilibrium reaches. The amount of water entering the cell is equal to amount of water leaving the cell. Turgor pressure develops in the plant cells only because of the presence of cell wall which is able to resist the pressure. It is a real pressure, not a potential one, and can occur to a great extent. In case of animal cells, the plasma membrane bursts if the pressure increases. Turgor pressure plays a very important role in plants. Turgor pressure helps in maintaining the shape and form of the plant. The stem of herbaceous plants and the one with non-woody tissues like maize, sugarcane and banana are held straight by fully turgid cells packed tightly together. Turgor pressure holds the leaves in a flat and horizontal position by keeping the mesophyll cells turgid. Turgor pressure helps in cell enlargement and consequently in stretching of the stems. Opening and closing of stomata is governed by turgidity of the guard cells. Certain plants like bean, nut plant, Mimosa pudica, show quick response of leaves by turgidity. This is a turgid cell showing osmotic pressure, turgor pressure and wall pressure. Availability of water in the soil. The plants absorb water through the root hairs from the soil. The soil contains water in forms. Gravitational water. It is the water that drains downward through the soil. The level to which it drains is called the water table. The water table of a place differs in depth due to rainfall. The gravitational water lies far below and is generally not available to plant roots. It is of extreme importance as it causes washing out of minerals and nutrients from the soil, called leaching. Part of water that is retained by soil could be hygroscopic water and or capillary water. Hygroscopic, it means it is the water that is retained as a thin film around the individual soil particles. Strong attractive forces between the soil particles and the water molecules hold this water tightly. This is the water least available to the plant and is generally the water left in the dry soils. In the clay soil, it amounts to about 15% and in the sandy soil to about 0.5%. Capillary water, the soil particles always have very fine pores in between, forming a very fine capillary system. As the water spreads, it fills the finer pores and is held around the soil particles by capillary forces against the force of gravity due to high surface tension of water. It is this water which is readily available and is easily utilized by the plant roots. The clay soil being very fine textured holds much more water than sandy soil. When a soil is watered, it retains good amount of capillary water and this condition is known as capacity, absorption of water. Major portion of water required by plants is absorbed by roots, but in some cases water may be absorbed by leaves and stems also. Root hair is a specially modified epidermal cell meant for absorption of capillary water of the soil. The plasma membrane and the vacuolar membrane tonoplast act as semi-permeable membrane and water is absorbed by osmosis. Soil solution should have a higher water potential as compared to root hair cell. Then only water will enter the root hair cell. Once into the root hair, water will pass into cortical cells, endodermis, pericycle and into the xylem vessel. The movement is purely dependent on water potential gradient. Water movement into the plant follows two pathways, symplast and apoplast. Cytoplasm of the entire plant is connected through a plasmodesmata strand, forming the symplast system. Water movement through the cells takes this symplast pathway by osmosis. The cell wall and the intercellular spaces form the apoplast pathway, which allows water movement inside the plant by the phenomenon of capillary and adsorption. The water absorbed through the roots is transferred radially to the xylem from where it reaches to all the other plants of the plant by vertical conduction of water through the vessels. You can see this in this figure. Apoplast pathway, symplast. Next diagram also. So conduction of water through the xylem. The content of xylem vessels is known as xylem sap. Various theories have been postulated to describe the lifting of the xylem sap or ascent of sap in the xylem. Root pressure theory. If a stem is cut few inches above from its base with a sharp knife, xylem sap is seen flowing out through the cut end. This phenomenon is known as exudation and this is due to the positive pressure developed within the root system due to continuous absorption of water by osmosis, which develops a positive pressure known as root pressure. This pressure can be measured and ranges from 3 to 5 atmospheres. But this pressure is enough to raise water to small heights in herbaceous plants. Physical force theory or cohesion theory. This theory takes into account of physical forces which act in case of very tall trees and water lifted up to great heights. The three forces that act together are force of cohesion. Cohesion means attraction between water molecules. Force of addition. Addition means attraction between water and lignocellulose walls of xylem. And transpiration pull, which lifts the water column by creating a tension inside the xylem vessel. Water forms an unbroken column starting from the intercellular space of the leaf mesophyll to the xylem of the leaf. Through stem and root the water in the soil. A water potential gradient exists between the leaf to the root, and transpiration causes a pull of the entire water column. So long as the column is an unbroken one from the outer atmosphere through the plant up to the soil, water is lifted up by the force of transpiration. You can see this in diagram. Compare them. Translocation of organic solutes. Movement of organic and inorganic solutes from one part of the plant to another is known as translocation. In simple terms, transport of sugar in sieve tubes is called translocation. There are experimental evidences to suggest that phloem is the tissue involved in translocation of products of photosynthesis, sugars. Sugar is produced in photosynthesis in the leaves and then sent to all parts of the plant for the growth and development of the plant. Leaf is known as the source where the food is produced, and all other parts of the plant which receive this food is known as the sink. Sink can be root, stem, fruits and storage organs like tubers, bulbs, rhizomes, etc. Thus, unlike conduction of water in xylem, which takes place in one direction from the root to a bud in the aerial part of the plant, phloem translocation from a leaf takes place in all directions. Mechanism of translocation. Sugar solution in the phloem. Sieve tube move along the water potential gradient created between the source and sink cells. Here there is a mass movement of sugar solution from the leaf mesophyll to the plant. Transpiration. What is transpiration? The loss of water from aerial parts of the plant in the form of water vapor is termed transpiration, and in the form of liquid is termed guttation. Transpiration may occur through three main sites in the plant: cuticle, lenticels, and stomata. Cuticle is the wax-like covering of the epidermis of leaves and herbaceous stems. Though it is meant to check transpiration, still about 10% of the total transpiration may take place through cuticle and known as cuticular transpiration. Lenticels. Lenticels are areas in the bark of a tree which are made up of loosely arranged cells, and about 0.1% of water loss take place through it. It is known as lenticular transpiration. Stomata. Stomata are minute pores on the epidermis of leaves whose opening and closing are controlled by guard cells. About 90% of water loss from plants takes place through stomata, known as transpiration. You can see this in this animation. Difference between transpiration and guttation very clearly. These are the two processes regarding water loss. So water is lost in the form of vapor, but in guttation, water is lost in the form of droplet, and it is also known as exudation. But in transpiration, the water vapor consists of only pure water, but in guttation, this water contains various mineral salts in the solution. Next one is water vapor is lost through stomata, lenticels and cuticle, and water is lost through pores. These are called hydrathodes, found at the ends of the veins. Transpiration may lead to the loss of turgidity in leaf cells to cause wilting. It has no effect on the turgidity of the leaf. So stomatal transpiration is regulated by the guard cells. The opening of hydathode cannot be regulated. Transpiration occurs in the presence of sunlight, and guttation occurs at night or in the early morning. Transpiration has a cooling effect on the plant. No such effect on the plant. So transpiration occurs during the dry conditions, and it occurs in the with conditions. Mechanism of transpiration. Transpiration occurs in two stages. Evaporation of water from the mesophyll cells into the intercellular spaces. Diffusion of this water vapor of the intercellular spaces into the outside atmosphere when the outside atmosphere is drier. Factors affecting transpiration. There are many external and internal factors that affect the process. Temperature. The increase in temperature increases the rate of transpiration by increasing the rate of evaporation of water from cell surface and decreasing the humidity in the atmosphere. Wind velocity. The increase in wind velocity increases the rate of transpiration by removing the water vapor of the atmosphere and lowering the relative humidity. Light. Light has got no direct effect in the rate of transpiration, but indirectly it affects the rate in two ways. Firstly, by controlling the stomatal opening, and secondly, by affecting the temperature. With increase in intensity of light rate or transpiration increases because stomata get opened and the temperature increases. Water supply. Deficiency of water supply in the soil decreases the rate of transpiration by decreasing the rate of absorption. When the deficiency of water in the soil becomes too much, then the plants with and do not recover from wilting unless water is supplied in the soil. This is known as permanent wilting. When in a hot and dry summer day, the plant transpires more than the roots are able to absorb, even though there is enough water in the soil. It is known as temporary wilting, as the plant recovers from such wilting in the late afternoon night. Atmospheric pressure. Reduction of atmospheric pressure reduces the density of external atmosphere, thus permitting more rapid diffusion of water. Plants growing on high will show higher rate of transpiration, hence they develop xerophytic characters. Atmospheric humidity. It means the amount of water vapor present in the atmosphere. The diffusion and evaporation of water depends on the vapor pressure gradient or the difference of water potential gradient between the atmosphere and the inside of the leaf. More the difference, more will be the rate of transpiration. Internal plant factors. Certain plant adaptations reduce transpiration. Reduced size of the leaves, thereby reducing transpiring surface. Some xerophytic plants have needle-like or spine-like leaves, spinous and open shear. Thick deposition of cutin, wax-like substance on the leaf surface. Stomata found sunken in the cavities, surrounded by hairs as in nerium and cycas. Root-shoot ratio. When there is more root and less of shoot system or leaves, there will more of transpiration. Root is the water absorbing surface and shoot or leaves is a transpiring surface. High root-shoot ratio will cause more transpiration. Role of stomata in transpiration. Since most of the water is lost through stomata, plants regulate the degree of stomatal opening and closing to reduce the water loss. It has been seen that stomata show periodic opening and closing during the day depending upon the heat and light, water content of the cell and humidity. They are generally closed during the night. From early morning till midday, the stomata are open and hence the transpiration increases till midday. During sunny afternoon, the stomata are closed and hence the transpiration decreases. From late afternoon till evening, the stomata are open again and hence the transpiration increases. At night the stomata are closed and hence transpiration is very low. Structure of stomata. Each stomata consists of a minute pore called stoma surrounded by two guard cells. The stoma acts as a turgor operated valve which closes and opens according to the turgidity of guard cells. The guard cells have unevenly thickened walls. The cell wall around stoma is tough and flexible and the one away from stoma is thinner. The shape of guard cells differs in dicot and monocots, though the mechanism remains the same. Mechanism of stomatal action. The opening and closing of stomata depends upon the turgor pressure in the guard cells. When the guard cells are turgid, the stoma opens, and when guard cells lose water, stoma closes. The mechanism of dicot and monocot is as given below. Dicot plants have kidney-shaped guard cells. The inner wall around the stoma are thicker than the outer wall. The guard cells expand, tough inner walls, stomata open, get distended by become. In monocot, the guard cells are dumbbell shaped. You can see this in the figure. When the guard cells region with thin and thick wall cells, stoma opens, become turgid walls bulges and gets moved apart, inflated. When the guard cells, the inflated part says, the thick wall, stoma close, loss of water, collapse. Changes in turgidity bringing about opening and closing of stomata has been known for a long time, but the mechanism that leads to turgidity needs to be unexplained. Starch sugar hypothesis. This hypothesis goes by the basis that the increase in sugar concentration due to photosynthesis and hence end osmosis of water during the day leads to opening of stomata, and the reverse. Decrease in sugar concentration followed by exosmosis leads to closing of the stomata at night. The changes in guard cells during the day, in light and at night in the dark are as given below. Reaction in the light, utilization of CO2 during photosynthesis. Drop in CO2 leads to increase in pH or protoplasm becoming alkaline. Conversion of starch into sugar, increased concentration of solute, end osmosis of water from other epidermal cell to the guard cells, increased turgor pressure of the guard cells, stomata open. Reaction in dark, accumulation of CO2 due to absence of photosynthesis, increased acidity or decrease in pH due to formation of carbonic acid, conversion of sugar into starch, decreased concentration of solute, exosmosis of water, decreased turgor pressure, stomata closed. So this theory cannot explain stomatal movement where starch is absent in the guard cells or guard cells lack chloroplast and opening of stomata at night and closing by the day in some plants like succulent. So effect of potassium ions on stomata. It has been convincingly proved that the accumulation of potassium ions brings the opening of stomata and loss of potassium ions the closing of stomata during daytime. Accumulation of potassium ions by the guard cells, increased solute concentration, end osmosis of water, increased turgidity. Wait a minute. So thus preventing an excessive water loss under experimental conditions. Also, when abscisic acid is applied to the leaves, guard cells close and check water loss. Significance of transpiration, absorption of water. Transpiration influences the rate of absorption of water from the soil. Water movement. By transpiration, water moves upward and as it passes into the cell vacuole, it makes the cell turgid. This gives a form and shape to the cells and to the plants as a whole. Mineral salt transport. The water stream moving upward also carries the dissolved minerals required for the development of the plant. Transpiration also helps in distributing these minerals throughout the plant. Cooling, the evaporation of water during transpiration cools the leaves. Protection from heat injury, some plants like cacti retain water by reducing transpiration. This saves the plants from high temperature and strong sunlight. So transpiration is a necessary evil. Stomata remain open during daytime for the absorption of carbon dioxide and release of oxygen for a very important process, photosynthesis. When the stomata remain open for this important gaseous exchange, escape of water vapor cannot be controlled. Thus, loss of water is a wasteful process which cannot be avoided because stomata must remain open to do something more important, that is absorption of carbon dioxide during daytime for photosynthesis. It is for this reason Curtis in 1926 has referred transpiration as a necessary evil. Factors affecting stomatal movement. Any condition which causes turgidity of the guard cell will cause stomatal movement. Solute concentration of the guard cell which will allow movement of the water into the guard cells. Turgid. Light causes photosynthesis in guard cells by chloroplast and hence accumulation of sugar in the guard cells. Concentration of potassium ions of the guard cells. Antitranspirants. Many crop plants give poor yield in dry seasons as the water lost by transpiration is much more than the water uptake by the roots. The rate of transpiration can be reduced by the application of certain chemicals known as antitranspirants. These chemicals should not affect the CO2 uptake. The reduction in transpiration is achieved by two means: chemical like phenyl mercuric acetate, PMA, and abscisic acid. ABA cause partial closure of stomata, checking transpiration to some extent. Some waxy substances like silicone emulsion form a thin film over the leaf and cover the stomata without affecting the uptake of CO2. Guttation. It is seen in early morning in the form of drops at the margins of tips of leaves of herbaceous plants. The plant in which transpiration is low and the root pressure is high. The liquid water is seen at the vein ending. It occurs through specialized pore called hydatodes present near the vein ending. It is quite common in young grass seedling and in the tropical rainforest due to warm and humid night. Tomato and nasturtium are some examples. Just see figure. So difference between transpiration and guttation. In transpiration, water is lost in the form of water vapor. In guttation, in the form of drops. Transpiration occurs through stomata, cuticle, and lenticels, but guttation occurs through hydatodes. So please compare this table. So what you have learned in this chapter? The movement of water from one cell to another depends upon the water potential of the cell. Water always moves from a region of lower solute concentration to the region of higher solute concentration along the water potential gradient. A more concentrated solution has a higher osmotic potential. Osmotic pressure is expressed in terms of energy. Water always moves from a region of high free energy to a region of lower free energy. Water potential is the capacity of a solution to give out water. It is represented by the word psi. It is affected by the solute concentration and external pressure. Psi is 0. More solute means low water potential. A solution has lower water potential than pure water. Water potential of a solution is a negative number less than 0. Plants absorb water by their roots from the soil through osmosis. The increased water content inside the protoplasm exerts a turgor pressure on the cell wall. The equal and opposite force exerted by the cell wall is termed as wall pressure. Water is present in the soil as gravitational water, hygroscopic water, and capillary water. The water absorbed by root hairs flows to the xylem vessels mainly by the Apst pathway. The water moves up xylem vessels to the leaf along the water potential gradient as explained by the cohesion tension theory. Transpiration or evaporation of water from the plant through stomata causes a pull and water moves up like a water column due to the force of cohesion and tension created by transpiration. Certain plants show guttation due to high root pressure and low transpiration. Turgidity of guard cells is explained by the increased conversion of starch into sugar and by the accumulation of potassium ions. Various environmental factors like temperature, light, wind, humidity, and internal factors like structure of leaf and root-shoot ratio affect the transpiration. Transpiration not only brings about ascent of sap but also has a cooling effect and saves the plant from injury. These are the some practice questions you have to solve yourself. Now we will meet in our